Hello, in this set of slides we will talk and describe a little bit about bacterial organisms. These are microbes that are prokaryotes, and we want to know more about them and so how they can fit into the soil environment. As we said, bacteria, they are mainly into the group of prokaryotic cells. They don't have true nucleus. Their DNA is swimming in the cytoplasm with known proteins. There are few internal structures. It is often unicellular, and the cell size is comparably small to those of eukaryotes. We call them the true bacteria. And examples of bacteria can be actinomycetes and cyanobacteria. While eukaryotic cells, they're different from eukaryotes because the eukaryotic cells usually have nuclear membrane, they have multiple chromosomes, they have several organelles, often multicellular and differentiated, they're large size, and for example, they can be grouped into like fungi, plants, and animals. For the description or the classification of microorganisms, and namely bacteria, we often rely on some phenotypic characteristics. Some relate to morphology, such as shape, size, and motility. Other physiological phenotypes, like the sources of carbon they require, what energy sources and inorganic nutrients that they utilize, and whether they rely on oxygen or not. In terms of biochemical analysis, we often consider the nature of the cell wall composition, their membranes, their cellular constituents. And when it comes to ecological differentiation, it helps sometimes to know a little bit about the habitat of the microbe and to be able and where it is isolated from, so we can give a little bit about how can we classify them and characterize them. For the phylogenetic classification, this is often more accurate and more precise. We use the ribosomal RNA as a conserved genes. For prokaryotes, we know that we use the 16S ribosomal RNA genes, while in eukaryotic cells, the story is a little bit different. We use the 18S ribosomal RNA gene. And the development of the phylogenetic classification was very helpful, since 1 to 10% only of bacteria in the soil can be captured on culture. So about 90% or more of the soil microorganisms and soil bacteria are often non-culturable. And with the aid of molecular technology, we've been able to isolate and characterize microbes that we cannot culture on the petri dish, and which gave us a little bit more about more diverse group and in the soil environment. So the genetic analysis gave more information about other non-culturable organisms. Well, when it comes to shape and function, we have mentioned before that bacteria there are several thousands of different species of bacteria, but they often share very few structural diversity. There are only 4 shapes of most bacteria. They could be either rod shape or bacillus shape. They could be sphere or coccus shape. They could be spiral or filamentous, and they could be vibrio shaped. So it's not very diverse in terms of structure. But if we can correlate between structure and function, or what habitat they could enjoy, for example, let's look at the rod-shaped bacteria. Usually their width is half about their length, and it comprises about 75% of soil bacteria. What is special about this shape that they have higher surface area to volume ratio. So there's more surface area to acquire water and other nutrients, which is helpful in the soil environment to allow the bacteria to capture more nutrients and water for survival and growth. While in spherical shape and cocci shape, they enjoy a greater structural strength than rods. They're not easily broken down. There's less distortion Their cells are less prone to distortion with drying. Therefore, they could survive more severe desiccation periods and higher pressure. They can withstand higher pressure than rod-shaped bacteria. So sometimes the rod has an advantage of being in the soil system because it can provide the cell with higher surface area to allow exchange of nutrients and so on. But in the cocci shape, especially for those in the water system, they're usually able to survive more severe desiccation and therefore have less surface area to volume ratio. And of course, they can have higher structural strength. The other bacterial shapes, as we mentioned before, it's like the spiral shape. This is not very common in soil. It's more in the wet sites because the spiral shape gives the bacteria ability to move and become motile, and it is more of submerged soils or mostly wet environments. The filamentous shape, also they have thin tubular structure called the hyphae, where most actinomyces and arthrobacters they belong to the filamentous shape. And the filaments give the bacteria the ability to move towards a nutrient or water and to acquire growth requirements. The chemical composition of mostly like bacteria or fungi compared to, let's look at the percentage of nitrogen. Mostly prokaryotes have higher nitrogen content, where 10 to 15 than fungi. And while in fungus they have higher carbon, where it's like more 10 to 15 carbon to nitrogen ratio. And in general, if we look about the contribution of bacteria and fungi in the biomass in soil, we can see that they're very similar and they can contribute to high biomass, and then therefore if the fungal or the bacterial cell dies, it adds up to the organic matter pool in the soil system, adding to carbon and nitrogen and perhaps other nutrients like phosphorus. The cell wall composition in prokaryotes is composed of a peptidoglycan sugar layers, amino acids, and it is only in prokaryotes. The cell wall is a little bit different from that in fungi, in eukaryotic cells. So if we look closely to the bacteria, for example, we can differentiate between two taxonomy, the gram-positive ones and the gram-negative. The gram-positive has thicker peptidoglycan. It's about 80% of peptidoglycan, while in gram-negative, it only has 5 to 20% of those peptidoglycan layers. So it has thin layers. And we could see here that they are a little bit, let's pinpoint some of it here. And this is the plasma membrane in this area here for both gram-positive and gram-negative bacteria. This is the cellular membrane here and here. Okay, and now the cell wall in gram-negative bacteria comprises of the layer of peptidoglycan over here, and then there's an outer membrane that surrounds the cell. While in the gram-positive one, we could see a thick peptidoglycan layer, and that's going to be useful for us later on to talk about the gram staining and how we can differentiate between these two groups. We have gram-positive and gram-negative bacteria, and these two are colorless when we first apply them into a slide to see them under the microscope. We do fixation as the first step to do gram staining. We add crystal violet to both of them, and they both stain purple. This stains purple, this stains purple. There's no difference so far. Then we add iodine solution. It fixes the crystal violet within the multi-layers of peptidoglycans. Then the most critical step in gram staining is the decolorization step, which is often in a shorter time. It is 10 to 20 seconds only. With ethanol, we can destain whatever crystal violet was inside the layers of peptidoglycan from gram-negative, since it has thin layer. But in gram-positive, it retains the stain because so much crystal violet is retained still within the layers of the peptidoglycan, because it has a thick cell wall. And then after applying the counter stain, the gram-positive will stay blue, but the gram-negative will turn out to be red, which is called the safranine stain. It has a red color because it has been already, the color has been dewashed from the gram-negative. It turns out to become colorless, and after that step, it can acquire the counter stain, which is the safranine. And this way, the scientist Christian Gram, he developed the first gram staining technique to differentiate between these two groups of bacteria, the gram-negative and the gram-positive ones. As we have mentioned before, the cell wall is important. It is made of peptidoglycan layers in bacteria. It provides rigidity and gives shape and resists osmotic pressure. The only known bacteria that doesn't have cell wall is Mycoplasma, and that's for the bacteria so far.